Hello everyone and welcome to this video where we discuss together one other aspect of the human physiology, and this time we are talking about the smooth muscle fibers. Now, just from looking at the slide, we recognize immediately that there are significant differences between the smooth muscles and the skeletal muscles, which we already went through over the lecture. And one of the aspects that we don't see the striation in the muscle fibers. Of course, it's a single nucleus, unlike the multi-nuclei present in the skeletal muscle fibers. In addition to that, the smooth muscles are not voluntary. You cannot contract and relax them as you wish, but they are under neural and hormonal modulation, as we will discuss later in this lecture. They do not attach themselves into the bones, into the skeleton. So these are the obvious differences between the smooth muscle fibers and the skeletal muscle fibers. And as we go through this lecture, you will realize that there are much more differences, although they're not as obvious, but they are very critical to the function and the way the smooth muscle cells are operating, and they do sustain their contraction. We'll talk about each of those aspects, but first just a general description of the smooth muscle cells. They are found in the walls of hollow organs and tubes, such as blood vessels, also the walls of your intestine, and you see them in the uterus, you see them in the fallopian tubes, around the pupil, the pupillary muscles or the iridial muscles. So you see them everywhere, and they don't have the arrangement of sarcomeres that we saw in the skeletal muscles, and that is why, of course, they lack the striation. Now, they're spindle shaped, as you saw in the previous slide. And the cells are generally arranged in sheets within the muscle. Now, those sheets can be circular sheets, and therefore they can reduce the diameter of that tube to the level that they can actually close down the tube if they act as sphincters, or they are arranged in longitudinal sheets, and that we see mainly in hollow tubes like in the intestine and in the fallopian tube, where they can aid in what we call the peristaltic movement. They have dense bodies, and those dense bodies contain the same proteins that is found in the Z lines of the sarcomeres, mainly alpha actinin. Again, most of the fibers are circular fibers. Like I said earlier, some gastrointestinal fibers can be arranged in longitudinal fashion, and sometimes we refer to the smooth muscle cells as the visceral muscles, that if we take the exception of the blood vessels and iris muscles of the equation, then we can start calling them the visceral muscles. The blood vessels can have a single layer of smooth muscles, which are present in the small arteries, but then when you go to major arteries, then you can see multiple layers of those sheets of smooth muscles. We have three types of filaments. That's unlike what we saw previously in the sarcomeres, where we talked mainly about two types of filaments. Here we have the thick filaments. Again, they're myosin, just like the myosin we had before, but it's arranged differently, and we'll see how it's arranged in a short bit. And we also have the thin actin filaments. And finally, we have what we call the intermediate filaments. And those intermediate filaments, they're not contractile, but they are part of the cytoskeleton framework that supports the cell shape and also transmits the contraction throughout the cell. Now, if we go to the thick myosin filaments, we will see that they are arranged in what we call side polar. And I'll show you in a very short bit what does that mean. Essentially, unlike in the sarcomeres, where the if you remember the slide of the sarcomeres, how the myosin heads were always arranged in the same direction, whether it's the upper or the lower. Here we will see that they are on the opposite direction of the thick filaments, and that allow the heads to move into opposite directions when they and then they will pull on the thin filaments into opposite directions, and we will see what that means in just a short bit. But for now, let's just remember this word, the side polar arrangement of the thick myosin filaments. Now here's a little depiction of how those filaments are organized. You will see that we have the thin filaments and the thick filaments. The thick filaments are those tiny parts here, and we will see better pictures in just a short bit, but I just wanted you to get a grip of the general arrangement of the muscles. If we consider this is one cell, one smooth muscle cell, we will see that we have the thin and the thick filaments are arranged in this fashion, and then they're tying themselves to what we call the dense bodies. Some of those dense bodies are within the cell, and some others are outside, associated with the membrane, and then they can also become a point of association using some connective tissue strands between one cell and the other. Therefore, we can transfer the contraction or unite the contraction between the muscle cells. So these are our thin filaments and thick filaments, and those are the dense bodies. Again, we will see better picture or better depiction in just a short bit. We don't see here the intermediate filaments, but the intermediate filaments, as I said, the intermediate filaments are these guys. So this is an intermediate filament. Here's those guys would be your intermediate filament, but it's not shown in this one except here and there. All right, so here's another histology picture for the smooth muscle cells. You see again their spindle shape, and they are arranged in sheets. This is an electron microscopy picture. What you see here is the lack of what we called earlier in the skeletal muscle. We don't have the sarcoplasmic reticulum. So most of the calcium is coming from an extracellular sources, and we have some crypts here around the outer cell membrane, and those crypts, they are full of calcium. We call those crypts calveoli, but we don't have the sarcoplasmic reticulum. And how does calcium enter to the cell? We will see in just a short bit. So, as I promised, this is a better picture for how the arrangement of the thin and the thick filaments. Those are the thick filaments here, and those guys are the thin filaments. The yellow oval here shape is your dense bodies, and when it contracts, it sort of causes also torsion of the cell, like a corkscrew. And as I told you earlier, that the arrangement of the myosin heads in those filaments, remember the myosin heads resemble like heads of a golf club, but the arrangement here is opposite to each other. So in other words, when those guys are moving, then they would pull this way, and that means we are pulling the actin filaments into this direction, whereas those guys on the opposite side, when they move, they will move in the opposite direction, and therefore I'm pulling the actin filament into this direction. And that's okay. The idea is that you're shortening your distances between your dense bodies, so you can shorten this one, but you can also shorten that, and they can this one can pull into that way, and the other one will pull into the opposite direction, and therefore we are bringing the two closer to each other, which is the idea of the contraction. Okay, so we learned before that the myosin heads have two binding spots on the head. One of them was for the actin, that was the actin binding area, and the other one was for the ATP binding or phosphate binding domain. So pretty much the same idea, but what you will see here is the contraction or the coupling between the myosin and the actin is not inhibited by the tropomyosin. Remember, in the skeletal muscles we had the troponin sitting on the tropomyosin, and the tropomyosin is blocking the myosin binding areas on the actin filaments. Now, we don't have that here, but we have other kind of proteins, and although it's not described well in your book, you can choose to forget this next part. We have here on top of the myosin head, we have what we call myosin light chain proteins, and those proteins are sitting here, and they would prevent the interaction between the myosin head and the actin. Now, those light chain proteins, the myosin light chain proteins, they can get phosphorylated, and we will see how they get phosphorylated. Once they are phosphorylated, they change the position, and that allows the myosin head to latch into the actin filament. We'll see how that happens, but just as a note, we do not have troponin in the smooth muscle fibers. We have tropomyosin, but the tropomyosin here, unlike the tropomyosin we had in the skeletal muscle fibers, does not play a regulatory role. The regulation between the myosin head and the actin filament is entirely based on those myosin light chain proteins I described for you, okay? Which are here, myosin light chain. So, if I would compare the striated muscles and the smooth muscles, in muscle excitation, we do have a rise in the calcium level, although the calcium is different, the source is different. It's entirely in the skeletal muscles from the sarcoplasmic reticulum, and you will see in my next slide that the calcium that goes into the cytosol varies a lot from its sources. Some of it is coming from the extracellular space and the calveoli, and some of that coming from intracellular storages, and we will see that in just a short bit. We know that in the skeletal muscles, the increase in the calcium would make conformational changes in the troponin, and that moves the tropomyosin away from blocking the myosin binding sites on the actin filaments, and therefore the cross bridge takes place. That we do know. Here it's a rather series of biochemical events, and I will describe that in the next slide. And whichever the reason is, once I uncover here in the skeletal muscle the actin filament, or over here I am causing changes in the light chain proteins, the idea is that you're causing the bridge, the cross bridge to occur, and that will cause the contraction in both. So anyways, calcium, wherever it's coming from, is critical, of course. And the other thing is that ATP is also critical, whereas here ATP was critical for phosphorylation of the myosin head. Here the ATP is critical for the phosphorylation of those light chain proteins. Okay, so this is the mechanism in a little bit more details. If calcium would enter, and we will talk how calcium does enter, it will bind to a protein called calmodulin, and the calmodulin has an enzymatic activity, and when the calmodulin gets its 4 calcium ions, it will change shape, and therefore it becomes active, and that will phosphorylate another protein called myosin light chain kinase, and that light chain kinase will phosphorylate indeed the light chain proteins, and therefore they are activated. This we do not see in the skeletal muscles. So I want you to remember the importance of this important protein called calmodulin, and the fact that the interaction between the myosin heads and the actin filaments in the smooth muscles is not related to the tropomyosin. It's actually related to the activation of calmodulin, and from there the activation of myosin light chain kinase that phosphorylates the myosin light chains, and therefore we have the cross bridge occurring between the thin and the thick filaments. Now, the smooth muscles, if we categorize the two, if we categorize the smooth muscles, we will see that we have something called multi-unit smooth muscles, and the other one is single-unit smooth muscles. Essentially what it means, the single-unit smooth muscles will contract all as a one unit. Like what? Like the sphincters, like the pyloric sphincter, for example. Everything is contracting together at once in one sensation. Whereas the multi-unit, they can choose the level of contraction. It can contract a little bit, or it can become relaxed, or it can. So there is sort of graded level of contraction that can happen in those smooth muscle fibers. The best illustration, because we see this illustration all the time, is in our eye, where the iris can respond to various degrees of light by closing. So it's not all or none. It's not like it's either open or closed, but it can close a little bit or a little more until it's a very, very small opening, like when you are in a very bright light out in the sun. Now that's very different than the single unit. In the single unit, it's either contracting or not contracting. We don't have various degrees. And the other thing, of course, other place we have the multi-unit, of course, is our blood vessels. Because you can imagine, if we want to fine-tune our blood vessel tone all the time, and we will study that in greater details when we get to the blood vasculature and when we get to the autonomic nervous system. You want a fine control. You don't want either your arteries are relaxed or they are tight. You want all the shades, all the gray shades. It's not black and white. We need much better control. And because we need all that fine control in various degrees, you will see that those type of cells have to be regulated by neurons. So we call that neurogenic type, because they will be regulated tightly by neurons coming from the autonomic nervous system, whereas the single unit, they are the signal, the contraction is initiated by the muscles themselves, and there are many ways to initiate that, but it's intrinsic and it's not exactly under the same control we see with the neurogenic, although we know, of course, that the autonomic nervous system can also play a role in accelerating or decelerating the motion of your smooth muscle fibers, even in a single unit such as your stomach or the intestine. And we will talk about that in greater details when we get either in the gastrointestinal tract or in the autonomic nervous system. But anyways, for now, let's please remember two different types of the smooth muscles. One of them is the multi-unit smooth muscles, and the other one is the single unit smooth muscle. Now again, the multi-unit smooth muscles the fibers, the units must contract separately, and each of those portions of the smooth muscle will be stimulated by nerves, and they are found in areas where I want graded contraction, not all or none, not black and white contraction. And you will see that in the areas of large blood vessels, the lungs, the muscles that adjust the eye lens, and we call that the ciliary muscles in the iris of the eye, even in the base of the hair follicles. All of these guys will belong to what we call multi-unit smooth muscles. Now, the single unit smooth muscles, they are self-excitable. It doesn't mean they're not regulated by nerve input. They are regulated by nerve input, but they're self-excitable, and you will see how they excite their own. And the muscle fiber contracts in what we call functional syncytium. All the muscle fibers in that particular circle, not in the whole organ, but in that particular region, in that particular circle, will contract together. And the contraction is slow, energy efficient, and we will talk about why are the contractions of the smooth muscle cells are energy efficient in just a short bit. Okay, so I described to you the importance of calcium, how calcium is critical. Why? Unlike in the skeletal muscles, please do remember that portion. Unlike in the skeletal muscles, where calcium binds to troponin and troponin moves tropomyosin from blocking the myosin binding sites on the actin filaments, here we have the calcium causing changes in what we call calmodulin, and the calmodulin will activate another protein, and that is the myosin light chain kinase, and we know the remaining of the story. But how is calcium entering the cell? Well, we have many ways. You can initiate calcium by stretching the cell. This is very unlike the skeletal muscles, where we did not have this mechanism. But over here, if we stretch the smooth muscles, we can allow calcium to come in from the extracellular space, and if calcium comes in, then it will cause contraction. So you see, it doesn't have to depolarize in order for it to contract. Matter of fact, as long as we get calcium inside the smooth muscle cell, depolarization is optional. It doesn't have to depolarize. Calcium alone can cause the contraction, the activation first of the calmodulin, and you know the rest of the story. Another place where we do not have depolarization is if it is by a ligand, and then we have a second messenger system where we are activating a receptor, and the receptor will activate a G protein, phospholipase C, PIP2, and then inositol phosphate 3, and that will allow calcium to come out from the sarcoplasmic reticulum within the cells. So that's another way where we do not have depolarization in order for calcium to come in. A third way also is not dependent on depolarization and does not require depolarization of the cell is when we have leak channels, and those leak channels will allow calcium just to seep in from the extracellular space, from the calveoli, and therefore when you have enough, then you will start firing contraction on your own. And that's very important because many of the muscles, like for example the blood vessels are on a constant level of tone, contraction. They have the contraction all the time. You can increase the contraction or you can reduce the contraction. If you increase the contraction, then you have stronger tone, and therefore your arteries are tighter, and therefore your blood pressure goes up. And or you can inhibit that tone, and therefore the walls of the arteries and the veins will relax, and therefore your blood pressure will go down. So we can indeed regulate that, but it doesn't mean that the contraction of the smooth muscle cells is always initiated by neural signal. No, it's always on, except for those multi-unit that we discussed earlier, like the ones around the iris and in other places. But even those are also in a state of contraction, but you can choose either you inhibit it or you can increase it. Now, how do you increase it? By increasing the calcium level. How do you inhibit it? By reducing the calcium level. But you can also induce or inhibit by playing with your calmodulin, or you can play with the myosin light chain kinase, and how long it's phosphorylated, how long the proteins are phosphorylated. There is another protein, there is another enzyme called phosphatase, myosin light chain phosphatase, that's supposed to remove the phosphate group and therefore inactivate those MLC proteins. And if you inactivate them, that will not allow the myosin heads to interact with the actin anymore. So at any event, calcium is very critical for the function of the smooth muscle fibers. So it's very understandable that many of the medications that we use for high blood pressure are calcium ion blockers, because we do not want the tone of the blood vessels to be extreme. We want to reduce it a little bit, and you can use calcium ion channel blockers for that purpose. All right, so whichever the reason is, whether it's by voltage operated, that means that depolarization here took place in order for calcium to come in, or we have stretch activated channels, or we have leak channels, or by a second messenger. Whichever the reason is, we can cause phosphorylation of calmodulin, and you know the rest of the story, what happens after that. All right, but remember also that exiting calcium from the cells is very important, because when you take calcium out of the cell, that's how you initiate relaxation. And interestingly, in the smooth muscle cells, we have two mechanisms how we can put calcium out. You don't have to know the details of that, but it's very economical. The smooth muscle cells does not require the same kind of energy that we saw in the skeletal muscles, and that allows the smooth muscle cells to operate for a very long time. You never see your arteries say, oh, you know what, we are tired, we have fatigue, we can't carry on. But your skeletal muscles, if you walk a distance or you run a marathon, then your skeletal muscles are giving up, because they can reach fatigue much easier than the smooth muscles. The smooth muscles are very efficient. Somehow some people talk about 200 times to 400 times more efficient when it comes to energy than what we saw with our skeletal muscle fibers. Okay. This is just a depiction of how the autonomic nerve fibers can communicate with the smooth muscle cells. And we see here, we don't see the motor end plate we saw with our skeletal muscle system, but we see what we call here varicosities, and those bulbs here will be full of neurotransmitter. Whichever the neurotransmitter is, it can be acetylcholine or norepinephrine, and those will be released into the muscles, the muscle cells, causing activation. What's interesting, and this is very important point of difference between the smooth muscle cells and our skeletal muscle system, is that between the muscle cells, between the smooth muscle cells, particularly in the single unit muscle, in the single unit smooth muscle, we will have lots of gaps between the cells, between this cell and the cell below, between this cell and the cell here, and that connects them electrically. If you remember, we call that gap junctions. So that will allow a signal, if it's induced here, to be propagated all over those muscles and the muscle cells, and therefore allowing what we call syncytium or organ syncytium, where all the muscle fibers that are connected together can contract at the same time without waiting for this. Of course, they don't all happen at the same time if you think of the esophagus, for example, but because it's transmitted as waves, you can start that those waves are moving from one point to another, and that causes the peristaltic movements. And we will see the peristaltic movement, a very interesting movie toward the end of this lecture about the peristaltic movement. All right. Gonna skip through this with the action potentials, and I'll just show you a few slides here to demonstrate to you how the smooth muscle fibers look like. This is from an artery, and you can see the smooth muscle fibers, how they are organized in this artery. And here it is from a uterus. It's much more complicated in the uterus, and we will talk about that when we get into the reproduction system and how they encircle, we call it the myometrium, how they encircle the blood vessels, and they are very critical in initiating the menstruation and also in stopping the bleeding. We will talk about that when we get more into our reproduction system. But think of it, think of the idea that the uterine muscles are not functioning in most of the times, and they only start to function during menstruation, sometimes during climax, and also during childbirth. They become very, very strong in order to cause the delivery of the baby. And we will talk about this later on. Here is from intestinal wall, and we see here two layers. This one will be the circular, and when we cut through it, then of course it doesn't look much of spindles anymore, and those are the longitudinal fibers, and you can see how again the spindle shape structure of those muscles look like. And please do remember that we do not have the striation here that we saw with our skeletal muscle fibers. So let's see this video together, which shows the peristaltic movement of the pyloric opening, and you can see how the pylorus, which is the opening, the upper opening of your stomach, the junction between the esophagus and the stomach, is sort of opening and closing, as you can see here in this video. That's with an endoscope, and I just wanted to demonstrate to you how the smooth muscles are contracting and opening and causing this peristaltic movement. Somehow the video is getting very pixelated for some reason. So that was one example. I just wanted to show you how the smooth muscles are contracting. The other example is much simpler, and that's from our iris and the pupil reflex. When you shine, I downloaded it from YouTube, of course, and when you shine a light, if it's in the dark, you will see that the pupil is open, and the more the light gets closer, the smaller the pupil will become. We will talk much more about this when we get into the eye, the physiology of the eye, and the special senses. But for now, I just want to show you also that there is gradual degree of closure. The more you shine the light, it will get tighter and tighter. So it's not all or none like we see with the single unit muscle fibers or the single unit smooth muscle. So points here that we need, now that we understand a lot about the physiology and the mechanism of contraction in the smooth muscle cells, some points here that we definitely need to remember after reading this chapter. As you saw, if calcium gets released into the cell, it has to bind to calmodulin. Calmodulin has to activate myosin light chain proteins, and the myosin light chain kinase, and the myosin light chain kinase will phosphorylate the myosin light chain proteins, and that will cause the cross bridges. So you can see many steps are occurring indeed between the time calcium came in to the time the cross bridge actually took place. So that causes the contraction of the smooth muscles to be much slower than that of the skeletal muscle. We don't have a sudden twitch. It starts gradually to build up, and that's because of the calcium. The more calcium we have, then the more the contraction, the stronger the contraction will be. So it's a very important point to remember that it's slower than the skeletal muscles. And why? Because we have many chemical reactions before we can get into the contraction to occur. Now, the contraction of the smooth muscles is very long lasting, as we have a phosphorylation now of these proteins, the light chain, the myosin light chain proteins. These guys do not get dephosphorylated easily. If you remember, the dephosphorylation was something automatic that happens in the myosin heads. We didn't need an outside source to cause the dephosphorylation. Here, indeed, we have another protein that is an outsider. It's called a myosin light chain phosphatase. And so it needs to be employed. It needs to take some time in order for you to dissociate the two proteins from each other. So that will take some time. The other factor is that calcium, in order to bring it back wherever we got it from, will also take some time. So whichever the reason is, it will cause the contraction of the smooth muscles to be a latching mechanism, not like what we saw in our skeletal muscles, where we had a 50 millisecond twitch that goes up, goes down, everyone goes home happy. Over here we have it different. Okay, now it's economical. In other words, it doesn't use much of the very valuable currency, which we called adenosine triphosphate. 1 ATP is enough for a latching between the myosin head and the actin for a significant long time. It's not like the twitches in the skeletal muscles, where we needed ATP all the time in order to cause the excitation contraction coupling and the cross bridge to continue between the myosin and the actin. We don't have this here. Indeed, we have the ATP can be very efficient. In 1 ATP molecule can cause the latch between the myosin head and the actin for significant long period of time. So back again, so it's slower than the skeletal muscle, and we discussed why. It's long lasting, it's economical, it doesn't use much ATP. That's why it's working all the time. Imagine if your blood vessels or your intestine requires too much energy in order for it to function, you will be very tired all the time, because you are burning all the ATP molecules just with your arteries or with your intestine. You can inhibit it at the muscle fiber level, because some of these varicosities we discussed, if you remember that picture I showed you, which is the junction between the autonomic nerve fibers and the smooth muscle cells. It can release a neurotransmitter that will cause a hyperpolarization, or it can cause the calcium to be released at a lesser level. It can block the calcium release. It can activate the phosphatase, and therefore you can indeed inhibit the interaction between the myosin and the actin filaments. So you can inhibit it. That's unlike what we saw before with the skeletal muscle fiber. We discussed how it's initiated by various mechanisms. We saw that the calcium can come in either by ligand, and the ligand will activate signaling transduction, PIP, phosphoinositol 3 kinase, and that will phosphorylate inositol phosphate 3, and that will allow the release of calcium from the endoplasmic reticulum. And then we have the leak channels that calcium can come in, that causes spontaneous depolarization, or not depolarization, but spontaneous contraction of the muscle cell. Then we also have the stretch mechanism. If you stretch a smooth muscle, then you can indeed cause the contraction. Another thing I really want to emphasize with the stretching of the muscle, you know, when you eat really a lot, you go to one of those all-you-can-eat buffets and you eat a lot and your stomach is so full, or, you know, you drank too much. Does that mean your stomach is not working anymore? No, it does. But you know, if this same stretching, like 2.5 times stretching of an organ, if this would have happened in your skeletal muscles, your skeletal muscle would not be functioning, because there will be no point of communication between your myosin heads and the actin filaments. But that's not the case in the stomach. The stomach will still function. Actually, it's stimulated even more when you stretch it, which is a very important thing, because you want your arteries to respond to a stretch inside them by increasing the tone. So it's a very, very important mechanism. But what I want you to remember is that you remember the length tension relationship we had in the skeletal muscles. If we overstretch a muscle, like when you put your arms behind your back, your power is much obliterated. We don't have that here. The skeletal muscles have the ability to stretch 2.5 times beyond their original length and don't lose any of its power, or you can reduce the length to half of its original length and still you have a contraction. It's a very, very important difference between the smooth muscles and the skeletal muscles. Okay, so the stretch, this is what I mean here by the stretch contraction or the stretch tension relationship, is much more accommodating when it comes to smooth muscles than when we compare it to the skeletal muscles. As we discussed earlier, the contraction of the smooth muscles is mostly spontaneous, but it can be also regulated up and down by several factors, and we discussed some of them when we were talking the autonomic nervous system. Some hormones like the oxytocin, which causes contraction of the uterus. You can take some inhalers if you have asthma, and in asthma you have violent contractions of the smooth muscles that are lining the bronchioles, and you can inhibit it, or you can regulate that contraction by different mechanisms. And the final point is the sources of calcium. Unlike the single source we had when we were talking about the skeletal muscles that was coming from the sarcoplasmic reticulum stores, here we have from multiple places. It can come from the extracellular sources or from intracellular sources. It can come from the calveoli, it can come from just leaky channels, and the extracellular calcium will start to seep in. At any event, we have a much more diverse way of getting the calcium into the cell. So that concludes our lecture about the smooth muscle contraction. I hope, or the smooth muscle physiology. I hope you will remember the differences between the skeletal muscles and the smooth muscles. In your book, you will see a very interesting comparison between the smooth muscles and the skeletal muscles. Don't spend too much time reading about the cardiac muscle cells yet, because we have an entire chapter dedicated to the cardiac physiology. So leave that for the lecture and we will indeed talk about it. Okay, I hope this lecture you will find helpful to sort of bring it home and memorize and understand the physiology of this very, very important tissue, the smooth muscle cells. Best of luck, and we'll see you in the next lecture. Have a good day.